Hello students, this is Ashta K. Sanuj, Assistant Professor, Department of Microbiology, EMA College of Arts and Science. Today we'll learn about cancer genes. So let's get started. So we have seen in our first video lecture that cell divisions are tightly controlled sequence of events. These events are dependent on proper level of transcription and translation of certain genes. When this process do not occur properly, or it is faulted, or there is some malfunctioning, that will actually help in the unregulated growth of cells, which will lead to cancer. Now, in this video lecture, we will be focusing on the genes which actually helps a normal cell to change into cancerous cell. The genes have been categorized based into two broad categories depending on their normal functions. So the genes, okay, I'll write it here. The genes have been categorized into two types based on their normal functions. And one is, okay, let me say proto-oncogenes, and other one is tumor suppressor genes. Okay. So these proto-oncogenes, normally they are present in our body and they very much act like a normal cell. But when there is an alteration in the gene, the proto-oncogene will become oncogenes. Okay, so in this way you can say a normal cell will become a cancer cell when there is an alteration of the gene. So these genes, which is oncogenes, they actually stimulate or enhance the cell division, and sometimes they also contribute to tumor growth by inhibiting cell death. Whereas when you talk about tumor suppressor gene, these gene protein product will actually direct or indirectly prevent cell division, and it will lead to cell death. So in one way we can say the oncogenes will mostly help in the cell division, whereas the tumor suppressor gene actually controls the cell death, that is apoptosis. Now in this video series we are going to focus upon oncogenes, and we will be talking about few examples of oncogenes. These oncogenes, the actually talking about the oncogenes, these oncogenes just need, there is another thing which I need to tell you, that it needs just a single defective copy, or you can say gene, and it does not require growth factors. Quite seems quite interesting. So when I say about these two things mentioned here, yeah, the single defective copy or gene is enough to make a proto-oncogene into oncogene, but it is actually not the same in case of tumor suppressor gene, and it does not require a growth factor. That means in this thing, you're just saying you get this is mother cell and father cell, him, F, okay, the daughter will have definitely both the genes, right? If suppose a father or mother's any one gene is actually altered or it has got some defect, this factor is alone for it to change proto-oncogenes to oncogenes, right? But this is not same in the case of tumor suppressor genes. Well, usually we need growth factors, right? They for the cell division to occur. But if a cell has changed to a cancer cell, it does not require any growth factor, and it will just start multiplying on and on and on, and it will start forming its characteristic feature of tumor, and it just goes on. Okay, so a cancer gene was, you have to know that the cell division is regulated by a sequence of events which actually depends upon the transcription and translation levels. So when the process does not occur properly, unregulated cells growth occurs, which will turn a normal cell into cancerous cell. Now, talking about the genes based on their normal functions, they have been classified or categorized into two types, that is, one proto-oncogenes and tumor suppressor gene. These proto-oncogenes act very much like a normal cell, and it will not have any malfunction or non-functioning part. But once it gets mutated, it can change into oncogenes. Oncogenes have the capacity to stimulate and enhance the cell division, and also sometimes it will help in tumor growth, that is, it will inhibit the cell death. On the other side, tumor suppressor gene, it is directly or indirectly, it is involved in the cell death, that is apoptosis. So talking about the oncogene, single defective copy of gene is enough for a proto-oncogene to get converted into oncogene, and it does not require any growth factor. Now let's see each one of them one by one. Before that, let me just erase all these things. Okay, and also let me change my color. Okay. Oh, why did I unback it? Okay, sorry. The first gene we'll be talking is about HER2 or new. It is also called as ERB, capital B. Okay, this is the first gene which we are going to see. This gene encodes the human epidermal growth factor receptor. Let me Let's write it. Receptor type 2. Okay, that is why HER, human epidermal growth factor receptor type 2. These genes are actually present in moderate level in normal cells. So as the name suggests, it is cellular response to growth factor. So this human epidermal growth factor receptor type 2, actually it is present in all normal cells, and the function of it is to it is a cellular, it shows a cellular response to the growth factor. Now consider, okay, this is your cell, and it has got the receptors. Okay, no, okay, now you have the growth factor coming here. Okay, it is going to come and bind to it. When it just comes and bind, this the cell division occurs. That is in the normal case, right? So if it is defective genes are present, what will happen? Instead of one receptor, there will be plenty of receptors which are present on the surface, and all of this growth factor is going to come and bind, resulting in multiple cell divisions, right? It is going to divide, and it is going to divide and divide. Though this is a normal case and this is a cancerous case. It was found out that 20% of human breast cancer cases, there was an elevated level of this gene. And these genes are present, these gene proteins are present on the surface of the cell, as I told you, and it will be helping in the cell division. Sometimes due to the gene amplification of this gene is also thought to alter the response to tumor to treatment, as well as the ability of the tumor to grow and spread. So this is very important gene. Actually, this gene may make a tumor more aggressive. So let me just put it down like this gene, it will actually alter the response to tumor treatments. And also, if there is a high level of this gene, it might make tumor aggressive. And one more thing is that it actually can has the ability to make cancer cell more sensitive to chemotherapy. Okay, it makes, if there is a high level, that is one good thing, which cancer cells more sensitive to chemotherapy. The drug, if you think it is an antibody plus this gene which is used, and antibody which is used is the monoclonal antibody. and the name is trastuzumab. Okay, I hope I actually pronounced it correctly. And what happens is, in this case, this is the treatment part, which I just mentioned here. So it actually, what it does is, let me just use a different pen, different ink. What it does is, normal case, as I told you before, you have a growth, we have a receptor for that. Okay, when this is going to come and bind, the division takes place. Now you have, when it is a faulty, you have a couple of receptors present, so the division is high. Cell division takes place at a higher rate. But in this case, if you, when your cells are muted, okay, what will happen? The drug will be going and binding to one of the receptor when, okay, I should use, I think it's conveyed, this is a receptor. Okay, and this part is the, okay, I think I'll just move. So this part is the drug, that is a monoclonal drug antibody which has been given. So it just binds. So what happens, there will be no space for the binding to occur, right? It will be present, but it cannot bind, come and bind here. That is the action of this. So when you talk about HER2 or NEU, okay, it codes for the human epidermal growth factor receptor type 2, which this is present, this gene is present at a moderate rate, and this, it actually helps in our cellular response to growth factor. Normally your cells will have the receptor for this, and this will come and bind to it and helps in the division of the cell. But once it is mutated, what will happen? Instead of one receptor, there are many more receptors which are found, and when it binds, it will help in unregulated cell growth. So, if you think 20% of the breast cancer in those cases, there was always noted that there is an increase in this particular gene. And one more thing, if you see a higher concentration of this gene, it can also do certain things. That is, it can alter the response to tumor treatment. Also, it can increase or what aggressiveness of a tumor. At the same time, one peculiar advantage of this is that sometimes, because in the case of high amount of this gene, sometimes the cancer become more sensitive to chemotherapy. And specifying about the treatment, it has to be mixed with the monoclonal antibody. It will not allow the binding of the genes to the receptor, so there will not be any cell division. Okay, now let's go to the next gene. The next gene will be RAS. It is a kinase signaling pathway. This gene products helps in the kinase signaling pathway. Okay, now this kinase signaling pathway, it helps in to control the transcription of the gene and regulate cell growth and differentiation. Now it has got an on and an off switch. When it is actually on, the RAS protein will go and bind to a molecule called GTP. And when it needs to be off, the RAS protein will go, actually has to break the molecule GTP. Okay, this is for cell division, and this is when it is not required, okay, in that case. So when there is an alternation in any of these genes, what is it is going to do? There will be no breakdown of GTP, right? When there is no breakdown of GTP, what will happen? It will be in a state called on. That means it is going to produce many cells, right? So it is going to go, there is no death phase here. So it is going to go get more and more cells are produced, right? And will lead to cancer. So this RAS gene products helps in the kinase signaling pathway, which are essential. They actually control the transcription of genes and regulate the cell's growth and differentiation. So you can just say like there is an on and an off phase. If it is on, the RAS protein will bind to the GTP and the cell division starts occurring. But when it needs to stop, this RAS protein will actually break the GTP, and then the whole process of cell division will be stopped. But if there is any change in or an alteration in the gene, what it does, it is not going to break. No GTP is broke or does not break. When there is no GTP getting broken, the overall overexpression, right? There will be an overexpression and amplification. And what will happen? So it is going to go for the growth, proliferation, and it will ultimately change a normal cell to a cancerous cell. So definitely in this case, it is not going to, actually the cell division depends upon many positive and negative signaling. And here only the positive signaling is going to enhance, where that's why the cell production division actually takes place. So actually for a to become a normal gene to become a cancerous, in the case of RAS gene, only one thing which is essential here is a point mutation, which can change a normal cell to cancerous cell. Then talking about the drug, um, the anti-cancerous drugs against this. So you have drug which is against this RAS dependent pathways. So they are targeting the pathways of this RAS gene. And if you see, there are many cancers where you can see an high elevated level of these genes, like pancreas cancer, where it is 90%. You have colon, where it shows about 50%. Lung cancer, around 30%. Then you have thyroid, bladder cancer, then liver, skin, kidney. In most of these cases, you can see an elevated rise in the level of RAS gene. So, and also one peculiar thing which I have read and it was interesting for me is that the RAS gene mutation actually is used to detect certain cancer. Okay, this mutation detects certain cancer. So it is in the pancreatic cell cancer. Actually, it is very difficult to diagnose. So identification of the RAS mutation in the DNA of the pancreatic cell, which is shed in the feces, may actually help the doctors to differentiate between the chronic pancreatitis and also the pancreatic cancer. So it is one way you can actually, the elevated level will help us to detect cancer, which is quite a positive side. So, about the RAS gene, it helps in the kinase signaling pathway, and it has two, like, on. When it is on, this protein is going to act on a molecule, and it will act on GTP molecules. Okay, it will bind on it, and if there is, and then the cell division takes place. And if the cell division has to stop, then it has to break the GTP in order to do it. When there is an alteration in this RAS gene, definitely what it is going to do, there will not be any off switch. That is, only cell division is going to occur. Now, once division occurs, there will be overexpression of this gene, and which will lead to cancer. So just a point mutation is enough for a normal cell to turn into cancer. In different kinds of cancer, they have seen that there is an elevated level of RAS gene. And one of the good kind of feature is that when it is high in concentration, it can be used to detect certain cancers, especially pancreatic cancer. Now let's move on with the next. Next is the MYC. Okay, now this acts as transcription factor. Then mutation in this gene is found in many different cancers, especially if I need to focus, that will be Burkitt's lymphoma, then B cell leukemia, and also some cases of lung cancer. So how these genes get they get activated by gene amplification and gene rearrangement. Gene rearrangement. This is going to activate this gene. Now, talking about the gene rearrangement, it is nothing but is a break and reunion, reunion of chromosomes. So this gene arrangement is nothing but the breakage or reunion of chromosome, and this process can involve large amount of DNA and affect many genes. The movement of the gene or a group of gene to a different location within the same chromosome or different chromosome leads to altered gene. That you have a chromosome which is present and it is getting moved to a different or to the different location of the same chromosome. In that particular case, it will lead to altered gene, and there will be overexpression of that particular gene. So when actually this gene or group of gene, you can say, they actually, sorry. They move to a different location where it is in the same chromosome, or it can be on a different chromosomes or different chromosomes. Okay, this will help in the overexpression of cell function. So one kind of gene arrangement which I think you will be familiar with is translocation. In the case of B cell lymphoma, there will be a translocation which is occurring in chromosome number 8 and 14. And as a result, what will happen? There will be an increased production of this MYC gene. When there is an increased so what will happen? I'll technically give you a rough idea of what's happening. So consider this is one chromosome. Okay, very bad chromosomes. Another chromosome. Okay, now let's just give me different colors. And a different color again. Okay, now what will happen when there is an exchange of this, or there is a rearrangement of these genes? This particular part shown here in green will jump over to this part, and this will come and join here, right? So this is what happens in the case of B cell lymphoma, okay? That is in the chromosome 8 and 14. Now, the amount of MYC protein in the cell is very, very important, because the activity of the, let me just write it down, the activity of MYC MYC gene is balanced. Should be in a very balanced state. Otherwise, there are other protein which will actually oppose the activity of other protein, which will oppose the other protein, which is going to oppose the activity of this MYC gene. Therefore, increase in either protein will actually offset. There should be a balance maintained. Okay, there should be a balance which has to be maintained between this MYC gene and the other protein. Otherwise, there will be an imbalance created, and it will affect the cell growth or cell division. That means it has to be a MYC. It has to be in a very regulated level, right? Regulated level. And if not, the activity of this will be inhibited by other protein. Okay, so it should be like in a regulated level. Both these proteins should be in the regulated level, or it is going to affect the, what, it is going to affect the cell division. So if sometimes it was found in certain studies that when there is an increase in this particular gene, it sometimes allows in the programmed cell death, which is called as apoptosis, right? That is, And sometimes what will happen, this will help in the when there is an activity, increased activity inside the cell, this is going to help the cell to undergo apoptosis. But there is another protein, write it down here, yes, which actually is known as BCL2. Okay, now this will go and bind in that particular scenario where it is very high, it will go and bind and it will hinder this property of MYC, that is, or programmed cell death, which is a good in the case of cancer. Okay, so talking about the make, it acts as a transcription factor. Mostly these are seen in this kind of mutated gene are found in Burkitt's lymphoma and B-cell leukemia and also lung cancer. The main defect is that they get activated by gene amplification or by the gene rearrangement. When you talk about gene rearrangement, that is, there will be a break and reunion of chromosome like translocation. Okay, and the level of this gene has to be maintained properly, because it may there is a chance that other protein will come and bind to it, and there will be balancing will be disturbed, as a result of which the cell division may be progressive. And when there is really high amount or high level of this MYC gene, it actually helps. Studies have found that it actually helps in apoptosis. So in that case, there is again a villain there that is called the BCL2. It is going to come and bind to this protein. Then the property of apoptosis gets cancelled and actually prevents it. Okay, now let's see the next. Okay, now let's see the next, that is a SRC. It is the first oncogene discovered. Uh, it is identified by cancer causing agents of RSV. What is RSV? Rouse sarcoma viruses. Usually infects chickens and other animals. Now this is a retrovirus. And it is also a tyrosine kinase. So what is tyrosine kinase? Actually, kinase is an enzyme which actually transfer phosphate transfer phosphate groups to target molecule. So when this actually transfers, there will be a change in the biomolecule structure. Biomolecule structure change, right? There will be a change in that, which actually gives signals to the nucleus, and then it will regulate the cell. Now the cell will become defected, then it's cancerous cell. Okay, so that is the activity of kinase. That is, it is going to add phosphate group to certain molecules. Now, once it gets added, definitely there is going to be a change in the structure. Now, that will actually send signals to the nucleus of the cell, which will actually regulate the cell division, such way that a cell will end up becoming a cancerous cell. Now, how it is going to do is, it is going to infect the cell cell, then it will insert its own gene into the cellular DNA. So again, there is an alteration. So all the process can occur and a cell can change into cancerous. So such thing was seen when it was injected into a chicken. Within 2 weeks, the chicken got tumor. And similar studies were also found in the case of humans. And there are 9 different SRC genes. By the way they are processing their mRNA produced by this 9 gene, they produce 14 different types of proteins based on the mRNA. So talking about the SRC, this is a first oncogene which is discovered, and it is actually identified as a cancer-causing agent of raw sarcoma virus. It infects chicken and other animals. It is a retrovirus, and it is a tyrosine kinase. That is, it has the ability to incorporate a particular group, that is a phosphate group, into the host. Now, once it gets infected, it is going to inject its own DNA, its own gene into the host and uh, into the cellular host, and the whole process, as we know, it is going to take place. And now the cell probably gets mutated and it is going to cause cancer. And these viruses are actually known as, okay, I forgot to mention it here. These viruses are actually known as acute transforming virus. Now, this the once it gets injected into a chicken, it is going to form a large tumor within 2 weeks, and cell to grow in abnormal manner also was found in the case of human. Now there are 9 different SRC genes so far they were identified, and based on the uh, processing of mRNA produced by this gene, there are 14 different proteins. Genes, most cell levels, overexpression, and all these things will actually kind of, it was mostly seen in breast carcinoma. There is a overexpression carcinomas. And also in human, let me write it here, human neuroblastoma and colon cancer and lung cancer. So there was an elevated level of this protein or overexpression of this protein. Now let's go to the next protein, gene. The next one is the telomerase. Next is the, write it here, telomerase. So usually DNA replication, the process of DNA replication, what happens is that the chromosomes, as the DNA process, it is going to shorten up, and to a stage when it is too short, it will end the replication, end of the DNA replication. That is what happens in normal case. Now, The shortening of the chromosome serves to limit the number of times any given cell can undergo division, right? So to only a particular stage, it as long as that chromosome has, like, once a division occurs, it is going to just break the both the sides, and it is going to shorten up. So, like, as it is going to proceed, they will reach a point where it cannot replicate, and that time actually the cell will undergo no further cell divisions. Now telomere shortened to a critical length, the cell is unable to replicate its DNA without losing its genetic material. That is what happens in normal cells. But when you see the cancer cells, what they do is they are going to produce, even if they reach at specific small, they can still actually continue the replication. That is with the help of the enzyme telomerase. Now, talking about this enzyme, this is an enzyme which is normally present during fetal development, but it is not present in adulthood. That is, the genes which are responsible for this enzyme are not expressed, so it will be absent. But in when a cell, normal cell, changes to a cancerous cell, in that time it will help those genes to get expressed, and as a result, what will happen? Telomerase, the enzyme, will be produced. So what it does is, when you have a chromosome during replication, it gets shortened up. Okay, it gets shortened up. Let's get shortened up. Okay, to a particular level. Suppose the level, it will stop DNA replication. But in case of cancer cells, there will be no shortening of this. Okay, it is going to continue, and it is going to continue, and it is going to continue. And that particular moment where the telomere will be shortened, or the critical length where the further replication cannot occur or cell division cannot occur, will be lost. So it will have a required length of chromosome with the help of telomerase, right? But certain, it is not that all cancer cells can produce it. Sometimes few cancer cells cannot produce the telomerase. There is another enzyme which is called as HTERT. Okay, this is actually a proto-oncogene which is present. Okay, so this also helps in the same way like telomerase. It is not going to actually break the length of the chromosome. So telomerase is nothing but, during DNA replication, the end of the chromosomes are known as the telomeres. They get shortened up during each cell division. Now the shortening of the chromosome serves to limit the number of times an given cell can undergo division. Now, telomeres shorten to a critical point or critical length, the cell is unable to replicate its DNA without losing its genetic material. Now, the center enters a growth arrest, so there will be no further division. This is what happens in normal cells. Now, in case of cancer cells, the ability to replicate without reaching a state of arrest can occur with the help of telomerase, and in few, with the help of HTERT proto-oncogenes. Okay, now let's see the next type, that is BCL2, that is B cell lymphoma gene 2. Now, these proteins are associated with membranes and membrane activity. This protein is mainly important to control apoptosis. Normal case, if there is a damaged DNA, it will undergo suicide. That is, it is going to kill itself as it cannot form any functional product, right? But in the presence of this protein BCL2, this will prevent apoptosis. So there is no cell death. Still, this damaged gene has been carried out. Now, what will happen? Definitely, the rest of the part is very familiar for you. There will be an expression, overexpression of this gene, and it leads to what? It is leads to continued division of mutated product, right? The damaged product will start, cell division will occur. Okay, that is what this gene does. And this actually contributes to metastasis. What is metastasis? That is the ability of spreading cancer cell to spread from one origin of origin, from the point of origin to a different organ. And this this apoptosis control is disrupted, and so when you actually use certain drugs, it will actually have a very negative effect because it is preventing that part. The main aim is to uh, actually to stop the cell growth and also to kill those cells which are already present. When some drug, when you have some genes which is preventing the death of these unregulated cells, and no, if you actually give any drug based on that, it will definitely will be of no effect, right? As it is not going to do. So now drugs are developed that it will downregulate the BCL2, this particular gene. Now then it will allow the anti-cancer drugs to work more effectively, even at a low dose. That is the treatment which is given in this scenario. And one such drug which is given is the antisense nucleotide. Drug name is Guinescence, is a drug which is given. So remember, as it prevents the death of the cell, what it is going to do, any death of the cell, any drug which actually uh, helps to kill the cell is of no use. So the drug has to be given first to lower this particular gene, and then you can give any anti-cancerous drug to work more efficiently. Now let's talk about the last gene, that is EGFR. It's called the epidermal growth factor receptor. Now this epidermal growth factor receptor, as you know, it is a transmembrane protein. So actually it has a portion outside the cell, then it crosses the cell surface and sticks out in the cytosol. Excuse me. So actually, to make it short, it actually functions like an antenna, cellular antenna. Now, binding of certain proteins to this EGFR causes two EGFR protein to stick to each other. Now, when they stick to each other, they form these joint proteins are called as dimer. These are called as dimers. The formation of dimer will activate the receptors and which actually causes the autophosphorylation. Now, what happens is that two copies of protein will add some chemical that is a phosphate to each other. So there will be an elevated level of EFGR, which will lead to the cell proliferation. And these kind of genes can be seen more in case of breast cancers, lungs, head and neck, and esophagus. That is, they're going to form. What happens is that when this binding of certain proteins, when you say proteins, it can be any epidermal growth factors or transforming growth factors. When they bind onto this EGFR, this causes two EGFR proteins to stick out each other. That is, the joint proteins, these two proteins are called as dimers. Now when this dimer is formed, it is going to activate this receptor. Okay, now activation leads to the autophosphorylation. And when these two copies of proteins are added, it will actually bring about a chemical change in the cell. That is, maybe it is going to add some phosphate group. Okay, so because of which there will be an elevated level of EGFR, and it obviously leads to the cell proliferation. So these kind of cancers are seen in case of breast, lungs, head and neck, esophagus kind of cancers. And here it is very important to know is that it helps. Too much of this activity will help in angiogenesis, and also it causes metastasis. Now the treatment, if you talk about the treatment, it is a monoclonal antibody that will be a tyrosine kinase inhibitor, binds to the portion of the extracellular portion of the EGFR and prevents the ligands activating the receptor, because it is the receptors which is actually making the problem here, right? That is, it is doing the autophosphorylation. So once autophosphorylation happens, it is going to it is going to alter the genes by the addition of the phosphate group. So if you here the drug is used, that is a tyrosine kinase inhibitor, which is actually present, it will avoid the activation of the receptor. Okay, thank you.